Welcome to the TOEFL listening section. In this part, you will hear one or more conversations in an academic or campus context, such as a discussion between a student and a professor or between two students. You may take notes while listening. Listen to part of a conversation. You will hear it only once, so listen carefully. After the conversation, you will answer questions about what you heard. Some questions will ask you about main ideas and details. Others will ask about implied meaning, speaker attitude, or how ideas relate to one another. Use both what is stated and what is implied to answer.
Good afternoon, Dr. Martinez. Thanks for meeting with me during office hours. I have to finalize my paper on autoimmune disease mechanisms, and I need to clarify several points.
Good afternoon, Ethan. Tell me which mechanisms you are unclear about. Autoimmune disease mechanisms cover a lot of ground, so we should focus on the central issues that explain why the immune system attacks healthy tissue.
First, I want to be sure I understand tolerance. How do central and peripheral tolerance prevent autoimmunity, and how do they fail?
Central tolerance happens during lymphocyte development. In the thymus, T cells that strongly bind self-peptides are normally deleted in negative selection. The AIRE gene helps thymic cells present tissue-specific antigens to developing T cells. If AIRE or negative selection is faulty, self-reactive T cells can survive.
So AIRE is crucial. What about peripheral tolerance for cells that escape the thymus?
Peripheral tolerance uses several mechanisms. Regulatory T cells suppress autoreactive cells. Checkpoint molecules like CTLA4 and PD-1 reduce activation. Antigen-presenting cells must give both antigen signals and costimulatory signals. If costimulation is lacking, autoreactive T cells become anergic or die. Breakdowns in any of these steps can allow an autoimmune response.
You mentioned costimulation. Is that why drugs like abatacept work in rheumatoid arthritis?
Yes. Abatacept mimics CTLA4 to block the CD28B7 costimulatory interaction. That reduces T-cell activation and downstream B-cell help. It is not a cure, but it reduces immune-mediated tissue damage.
I see. Another area I am fuzzy on is the role of genetics. How do HLA alleles influence autoimmune risk?
Certain HLA class II alleles present particular self-peptides more effectively. For example, HLA-DRB1 shared epitope alleles are associated with rheumatoid arthritis. Those alleles can present citrulinated peptides more readily, which increases the chance of activating autoreactive T cells.
You said citrulinated peptides. That is a post-translational modification. How does that lead to autoimmunity?
Post-translational modifications change amino acids in self-proteins, so the modified peptide is different from what the immune system saw during education. For example, citrullination converts arginine residues to citrulline and creates new epitopes. Those new epitopes can be perceived as foreign and provoke both T-cell and B-cell responses.
So these neoepitopes can escape central tolerance because they did not exist when T-cells were selected.
Exactly. That is one reason autoimmune disease can arise later in life after environmental triggers cause modifications.
Speaking of triggers, I often read about molecular mimicry and bystander activation. How do these compare as triggers?
Molecular mimicry is when a foreign antigen, such as from a virus, shares structural similarity with a self-peptide. An immune response to the pathogen can cross-react with self-tissue. Bystander activation is different. Infection causes inflammation that activates antigen-presenting cells and releases cytokines. This environment can activate nearby autoreactive lymphocytes that were otherwise quiescent.
Can you give concrete clinical examples that show these mechanisms?
One often discussed example is type 1 diabetes. Viral infections have been implicated in triggering autoreactive T cells that attack pancreatic beta cells through molecular mimicry or bystander activation. Another example is rheumatoid arthritis, where smoking can promote citrullination in the lung, leading to anti-CCP antibodies in genetically susceptible people with certain HLA alleles.
What about systemic lupus erythematosus and immune complexes?
In SLE, autoantibodies form against nuclear antigens. Those antibodies bind their targets and form circulating immune complexes. Deposition of these complexes in kidneys and blood vessels activates complement and causes tissue inflammation and damage. That is more of an antibody-mediated mechanism, rather than direct cytotoxic T-cell killing.
Are there distinct cytokine profiles in different autoimmune diseases?
Yes. H17 cells and IL-17 are implicated in tissue inflammation in diseases like psoriasis and some forms of arthritis. IFN-gamma and cytotoxic T-cell responses are important in type 1 diabetes. Type 1 interferons are prominent in lupus. Understanding these profiles help develop targeted therapies like anti-IL-17 and anti-interferon drugs.
You mentioned epitope spreading earlier. Could you explain that with an example?
Epitope spreading happens when the initial immune response to one epitope damages tissue and releases more self-antigens. The immune system then begins targeting additional epitopes. In multiple sclerosis, an early response to one myelin protein can broaden to include multiple myelin components as disease progresses.
How do current treatments address these mechanisms rather than just the symptoms?
Therapies target different points. B-cell depletion with anti-CD20 reduces autoantibody production. Checkpoint modulation with CTLA-4 Ig reduces T-cell activation. Cytokine blockade targets inflammatory mediators. There is also active research into antigen-specific tolerance therapies that aim to retrain the immune system to tolerate a particular antigen.
Are there any promising antigen-specific approaches in trials?
There are experimental vaccines that deliver peptides with tolerogenic carriers, or target tolerogenic dendritic cells, to induce regulatory T cells. Results are early, but the idea is to restore tolerance to disease-relevant antigens without broad immunosuppression.
One final question. Many autoimmune diseases have a strong sex bias favoring females. Do we know why?
Hormonal influences and sex chromosome-linked genes both matter. Estrogen can modulate B-cell survival and promote antibody responses. X chromosome genes involved in immune regulation can escape inactivation and contribute. These factors interact with genetic and environmental risks to increase prevalence in women for many autoimmune diseases.
This is very helpful. I will include central and peripheral tolerance, AIRE, HLA associations, post-translational modifications, molecular mimicry, epitope spreading, and cytokine profiles in my paper with the specific disease examples you gave.
Good plan. Make sure you explain how each mechanism leads to loss of self-tolerance, and use the examples to show clinical relevance. If you want, I can review a draft next week.
I would appreciate that. Thank you for the detailed explanations.
You are welcome. Good luck with your paper, and see me if you need figures or references.
Listen again to part of the conversation. Then answer the question.
Post-translational modifications change amino acids in self-proteins, so the modified peptide is different from what the immune system saw during education.
This concludes the conversation portion. For more TOEFL practice tests and study materials, visit Spinta.org.